Hello students. So today through this video I will be discussing about ISC semester 2 examination specimen question paper biology paper 1 theory. So here maximum marks 35 and candidates are allowed on additional 10 minutes for only reading the paper, and they must not start writing during this time. Now in this paper there are three sections: section A, section B and section C. In section A 7 mark, in section B 16 marks, and in section C 12 marks. Okay, so let's begin. In section A 7 marks, here the first question is, give the biological name of the causative agent of gonorrhea. And here the answer is Neisseria gonorrhea. This is the answer. This is the causative agent of the gonorrhea disease. Next, question number 2. Sonora 64 is a variety of wheat, rice, maize, sugarcane. So here the correct answer is Sonora 64 is a variety of wheat. Next. Next question. Assertion reason. PAN is a secondary pollutant, and reason, the secondary pollutants are produced by the combination of primary emitted pollutants in the atmosphere. And the options are both assertion and reason are true, and reason is the correct explanation of assertion. B. Both assertion and reason are true, but reason is not the correct explanation of assertion. C. Assertion is true, but reason is false. And D. Both assertion and reason are false. So secondary pollutant means those are not actually arised directly from any source. So it mainly produced due to the reactions between the primary pollutant. And here PAN, actually it is a secondary pollutant. So this assertion, it is correct. It is true statement. And the reason which is given, that these PAN, that means these secondary pollutants, are mainly produced by the combinations of primary emitted pollutants in the atmosphere. So here the reason which is given, this is also correct. So the first option, both assertion and reason are true, and reason is the correct explanation of assertion. It will be the correct answer. Here both assertion and reason are true, and reason is the correct explanation of assertion. Come to question number 4. Give one significant contribution of Professor R. Mishra. So actually Professor R. Mishra, he is very famous, and he is actually the main constructor of the Indian ecology, and we call him as the father of Indian ecology. Then next, so I have written here father of Indian ecology. Next question, expand the term ADA. Okay, expand the term ADA, and here the correct answer is adenosine deaminase. This is the expanded form of ADA. Next, question number 6. A substance that stimulates the production of antibodies is called dash, and here the correct answer is antigen. Here the correct answer is antigen. Okay. Question number 7. Why does a large ozone hole develop specifically over Antarctica? So here I am describing few things. The major ozone depleting substances are CFCs, and 14% they are responsible for total depletion. Nitrogen oxides are also responsible 3.5%, sulfur dioxide, halogens, carbon tetrachloride, methyl chloroform, chlorine. So these are some ozone depleting agents. Now chlorine liberated from the degradation of CFCs or chlorofluorocarbons, which mainly used in refrigeration. and it mainly released from the refrigerator and then it released into the stratosphere, and it is the main cause of ozone depletion all over the globe. So appearance of ozone hole mainly at the Antarctic is the result of action of CFCs. Mainly the main agent which is here depleting the ozone layer, that is CFCs. And under following conditions, that means the conditions which actually making it favorable for making the depleting of this ozone layer here: very low temperature. As you all know, Antarctic, it is one polar part of the earth and it is very cold area, so very low temperature, this condition. Then second condition, presence of stratospheric cloud which traps ice and ozone depleting gases, and presence of a polar vortex which provides a containment wall. So this is the answer of question number 7. So you can confine your answer here in this part, and the main agent is, or main depleting ozone depleting substance is CFC. And in the Antarctica, these three conditions are available, and due to that, in the Antarctica mainly we can see the depleting, much more depleting layer we can observe there in the Antarctica. Okay, so this is section A. Now come to section B, and it is 16 marks. So question number 2, briefly explain the role of the following in providing defense against infections in human body. Number 1, B cells, and number 2, T cells. Come to the answer. I have prepared everything here. B cells, these are also called as B lymphocytes. Okay, and they are short-lived cells of our immune system, and they survive only for a few days or weeks. And what actually they are doing for our immune system? They are responsible for antibody mediated immunity. Okay, antibody mediated immunity, this is the main function of these B cells. They are producing immunity through production of antibody. And for T cells, these cells have long lifespan, surviving for months or years, and they are responsible for cellular, this is the main answer, they are responsible for cellular immunity. Okay, so you just confine your answer in this particular part, that they are responsible for cellular immunity. T cells are responsible for cellular immunity, and B cells are responsible for antibody mediated immunity. Okay. Question number 3, what is the role of a probe in recombinant DNA technology? So I have written here, a probe is a single-stranded sequence of DNA or RNA used to search for its complementary sequence in a sample genome. The probe is placed into contact with the sample under conditions that allow the probe sequence to hybridize with its complementary sequence. So this is the role of probe in recombinant DNA technology or in genetic engineering. Question number 4. The level of air pollution is very high in a city. Suggest any 4 measures which should be taken by the government to control it. So these are that 4 steps that government can be taken for controlling the air pollution in the mainly in the major cities, where the air pollution is a very major problem. So first is switching over the entire fleet of public transport from diesel or petrol to compressed natural gas, that is CNG. This is the first controlling measure. Second controlling measure, phasing out of old vehicles. Third, use of lower sulfur petrol and diesel. And fourth, use of catalytic converters in all vehicles. Question number 5. If 20,000 kilocalorie energy is available at the level of producers means green plants, then what will be the amount of energy available at the level of secondary consumers? Now, if you observe the trophic level, producers are in the trophic level 1. Okay, and in trophic level 2 primary consumers are present, and in trophic level 3 secondary consumers are present. So here they have given the question that in producer level the energy is 20,000 kilocalorie, and according to Lindeman's law, in each trophic level 10% energy is transferring to its next higher level. So let's see what will be the answer. So here producer, it has 20,000 kilocalorie energy in its body, and it will be transmit 2,000 kilocalorie, that is the 10% of 20,000 kilocalorie, to the primary consumer, and then from there, from the primary consumer, it will be transmitting 200 kilocalorie, that is the 10% of 2000 kilocalorie, to the secondary consumer. And they actually asked the energy level of the secondary consumer. So the correct answer is 200 kilocalorie energy. The number of mice in a laboratory was 100 on a particular day. After 1 year, their number increased to 120. Now calculate the growth rate in a population. So for that we need to see one equation. So they asked for the growth rate, and there is one equation you need to remember. So growth rate R is equal to Pn minus Pi by Pi into 100. So here Pn is 120. Okay, initially the number of individual, that means the final record, okay, 120 which we got, and the initial record was Pi 100. Okay, that means initially the population was 100 and now it increased to 120. Okay, so 120 minus 100 by 100 into 100, and if you count it, we'll get the growth rate will be 20%, and this is the correct answer. Okay, so the question which is given, calculate the growth rate in the population, it will be 20%. Give, question number 6, give any 4 advantages of mutation breeding. So 4 advantages of mutation breeding. Number 1, it improves morphological and physiological characters of cultivated crops. Number 2, it can improve the disease resistance of crop plants. Number 3, it can induce desirable mutant alleles in crop plants. And number 4, it can be used to improve the specific characters of a well-adapted high-yielding variety. So these are the 4 advantages of mutation breeding. Question number 7. Name and describe the technique that helps in separation of DNA fragments. So for DNA fragments, the technique which we are using, that is called agarose gel electrophoresis. Okay, so this is the name of the technique. So I have written here agarose gel electrophoresis. This is the main technique. Okay, so they have asked also that name and describe the technique. So that means we have to give the description. We need to give a description there. So for before that we should know one thing: the DNA fragments, being negatively charged, move towards the positively charged electrode or anode under an electric field. DNA fragments resolve according to their size. The separated DNA fragments can be seen on staining with fluorescent dye, that is ethidium bromide, and followed by exposure to UV radiation. Then DNA fragments form orange colored bands, and these bands of DNA are cut out from agarose gel and are extracted by a process called elution. These purified DNA fragments are used for constructing recombinant DNA or are amplified into numerous copies by polymerase chain reaction. So the main process is agarose gel electrophoresis, and here I have given one figure which is showing the fragmentation of DNA, the separation of the DNA fragments. Okay, and lastly we can visualize it under UV radiation, and it will be appear orange colored bands. Then question number 8, which molecules are called molecular scissors? So mainly the restriction enzymes are known as molecular scissors, And why are they called so? Okay, so restriction enzymes are mainly known as molecular scissors. They are also called molecular scissors as they cleave DNA at or near specific recognition sequences. Okay, and that is known as restriction sites. So this is the answer for this question. Question number 9. Differentiate between primary succession and secondary succession with appropriate examples. So primary succession, this one is for primary succession, it is for secondary succession. Primary succession, I have to differentiate it first. So when succession begins on primary bare area which has not been previously occupied by a community, okay, then it is known as primary succession. An example, a newly exposed rock area, sand dunes, new islands, deltas, shore or recent lava flow. Okay, this is the example for the primary succession. For secondary succession, when community develop occurs in a secondarily bare area from where community was removed and where nutrients and conditions for existence are already favorable. This is known as secondary succession. An example, cut over forest, abandoned cropland and plucked field. So this is the example for the secondary succession. So so section B is also over. Now come to section C. Question number 10. Study the graph given below and answer the following questions. So here they have given one relation. In the x-axis they have, in the y-axis they have given species richness, and in the x-axis they have given the area. Okay, now explain the ecological principle represented by the graph and write a mathematical expression for the same. And what will happen if the slope of the line B, this one, B, becomes steeper? So here, this diagram is mainly indicating the species area relationship. Okay, so German naturalist and geographer Alexander von Humboldt, while exploring the wilderness of South American jungles, found that within a region the species richness increased with increasing area, but up to a certain limit. The relationship between species richness and area turned out to be rectangular hyperbola. You can see that rectangular hyperbola for a wide variety of taxa, whether they are birds, bats, freshwater fishes, or flowering plants. And on a logarithmic scale, it is a straight line. So here is the equation: log s is equal to log c plus z log a. Okay, and here s is species richness. S is species richness, Z is slope of line or regression coefficient, and C is y-intercept while A is area. Okay, so regression coefficient Z has generally a value of 0.1 to 0.2 regardless of taxonomic group or region. Example, plants in Britain, birds in California, or mollusks in New York. Okay, so this is the answer actually for this question. The species area relationship is mainly showing through this figure, and the main chemical equation is this one: log s is equal to log c plus z log a. Come to the next question. Explain the steps involved in the process of plant breeding. So the main steps which are involved in the process of plant breeding: first, germplasm collection for variability. Second step, evaluation and selection of parents. Third step, cross hybridization between selected parents. Fourth step, selection and testing of superior recombinants. Fifth step, testing, release and commercialization of new cultivars. And sixth or last step, multiplication of improved seeds. So these are the sequential steps through which we can involve the process of plant breeding. Question number 12. The rDNA technology has provided a method to control the nematode parasite Milioidogene incognita. Explain the principle involved in this technique. So what kind of technique they are using, that is called RNAi. Okay, RNAi. So let's see RNAi. This is called RNAi or RNA interference. Okay, so many nematodes, roundworms live in plants and animals, including human beings. Okay, so a nematode Meloidokin incognitia, the question which they asked actually, infects the roots of tobacco plants and causes a great reduction in yield. A novel strategy was coined by Fire and Mello in 1998 to prevent this infestation that was based on the process of RNA. RNA takes place in all eukaryotic organisms as a method of cellular defense, and this method involves silencing or breaking of a species specific mRNA. So if we see the steps, the first step is the nematode specific genes were introduced into the host plant using agrobacterium as vectors. As you all know, in the recombinant DNA technology, we need the vector. So here they are using the agrobacterium as vectors. Next, introduction of DNA was made to produce both sense and antisense RNA in the host cell. Next, the sense and antisense RNA were complementary to each other, so they formed a double-stranded RNA that initiated RNAi. Next, thus cleavage of double-stranded RNA takes place. This is called initial step. Next step, each si RNA complexes with ribonucleases distinct from dicer to form an RNA-induced silencing complex, that is called RISC. Next, the siRNA unwinds and RISC is activated. Next, the activated RISC targets complementary mRNA molecules. The siRNA strands act as guides where the RISCs cut the transcripts in an area where the siRNA binds to the mRNA. This destroys the mRNA. This is called effector step. And lastly, when mRNA of the parasite is destroyed, no protein was synthesized. It resulted the death of the parasite. That means that particular nematode in the transgenic host. Thus, the transgenic plant got itself protected from the parasite. So this is known as RNAi technique, and through this technique, we can prevent the infection. Here, the infection which mainly made by one nematode, okay, that is Meloidignia incognitia, and this infection is making in a plant, okay, mainly in the tobacco plant. So by introducing this technique, we can develop the immunity or preventive protection, or mainly the protective energy protectiveness we can form here in the tobacco plants by using this RNAi, okay, against this nematode. Last question of this section. Consider the amount of organic matter at the different trophic levels in an ecosystem given below. So here they have given tertiary consumers 250, primary consumers 150, primary producers 100, secondary consumers 200. Okay, so on the basis of the data provided above, construct an ecological pyramid. Okay, we'll construct an ecological pyramid here. Common upon its nature, giving at least one example of the type of organism occupying each of the above mentioned trophic levels. So let's see. So this is the pyramid which I made, and it is one kind of inverted pyramid. So in the primary producer level 100 kg, primary consumer level 150 kg, secondary consumer level 200 kg, and in tertiary consumer level 250 kg. So if we see the same kind of example, we'll get there in the producers, the oak tree and herbivores, the birds, parasites, lice and bugs, and hyperparasites, bacteria, fungi, and actinomycetes. So it is actually forming a pyramid. Is the pyramid which mainly formed, and it is the inverted pyramid of number in parasitic food chain. So if we see the example here, this is the example of inverted pyramid of numbers, and in a parasitic food chain, the pyramid of numbers is reversed as the number of parasites are more than their host. Okay, so for example, a single tree supports a larger number of fruit-eating herbivorous birds, which in turn support a still larger number of parasites like bugs and lice, which was already shown in this diagram, which is representing one inverted pyramid. So you can take this one also as example as well as the diagrammatic representation. Okay. Now, in question number 13, they have given another question. They have given or, and after that, they have given on the basis of the demographic data of a country given below, construct an age pyramid and explain whether the population is stable, declining, or growing. So we have to we have to draw another pyramid, but it will be a age pyramid. Okay, and they also asked few question about the population, when it will be stable, declining, or growing. So let's see. So this is the diagram, and the diagram which we got, that is URN shaped kind of age pyramid, where the pre-reproductive they have given 10,000, reproductive it is in 15,000, and post-reproductive 20,000. So this way you have to draw this diagram, this age pyramid. They also asked one question, that when the population will be growing or stable or declining. So growing population or expanding population, it has high birth rate and low birth rate. Thus, there is more number of young individuals in the population. And when it will be stable, it has equal birth and death rates. Okay. Then when there is the equal birth and death rates, then the population will be stable. Such a population shows zero population growth. And when it will be decline, so when the death rate is higher than the birth rate, in this case the population of younger one is lower than the old members. So we are getting the same kind of result here. Okay, so this is about the last part of this section C, and through this video actually I am just trying to explain all the questions, okay, about this specimen paper of ISC biology paper 1. So hope you like this video. Please subscribe my channel. Thank you for watching this video.